And the fourth one is beta 2, that has become where the homology of the acyanonucleotide protein increase, is not sticking on the side anymore, but enters the inside of the protein. Goes inside and destabilizes the whole thing and can open. So, and we have discussed about this with Mikhail, and we are now going to that in more detail. So this is the way we think we can make prediction using multi-resolution procedure. And to go back to I think I mentioned yesterday, we are now work in progress now to do the same thing with what active yellow protein. This is the last thing I presented yesterday. We have these, and this is the work of Paul, who's here in the audience. We have this prediction for the activated state of this protein. The activated state is not known because experimental technique such as NMR has not been able to be solved completely. This is a very large sample, partially unfolded, so it's not trivial. It's populated only for milliseconds. So we have now taken the full ensemble at the corresponding level, and we have done the full reconstruction, and now we want to test the energy, and we are also for the unfolded and the folded state, and we want to test the average energy, the energy level that we obtain with the final calculation in this ensemble, and this is what we need to obtain to confirm this prediction. We need that the folded state of the protein, this one, need to be, with the chromophore in the trans configuration, need to be the ground state, the lowest possible state. After photomerization, so if you take the folded state but put the chromophore in the cis configuration, the protein is not kept at all there because it triggers the conformational change. So we want to have a large energy gap with respect to the ground state. And then this activated state that is temporarily populated before the recovery after the decay of the chromophore must be in between because the protein moves from here to here to here. So we hope that this is what is going to be. The problem, the technical problem is that there is a photomerization limitation, so there is actually a quantum effect, and also there is a deprotonation event. So it's not as trivial to compute this energy, and fortunately we have the expertise of a quantum chemistry group that can assist us in doing the quantum calculation. And these are the two preliminary results we have so far. So this is the empty state. This is a snapshot of one configuration in the activated state. We have a very large ensemble in the activated state. So what I'm talking about here is one, we want to obtain a full atom pictures of the configurations here, so in this state here. And so if you see, the main difference is the core of the protein, this beta sheet remains basically the same. There is the chromophore that is inside here opens up and is exposed, and this part of the protein becomes completely unstructured all over the place. And so but the core remains the same. And this is in agreement with preliminary NMR data from a truncated variant. Energetic estimates seem to point in the right direction, but we don't have the final numbers. So if we compare with experimental with the NMR structure being published for a truncated variant, as I said, so it's not exactly the same, but it seems to be the truncated variant is much more stable. It's in a very good agreement. This is the NMR ensemble, the native, this is the native, the chromophore inside. This is one representative of our activated state, and this truncated variant indicates exactly the same thing. Sorry, the colors do not match, but this beta sheets remain the same. This part is completely unstructured, and this part is open up. Anyway, this is just, I hope to be able to tell you the rest of the story soon. So let me skip this. One thing I wanted to discuss. How much time I have? 10? Okay. one thing I want to discuss is that I keep telling you that it's very important to pick the right reaction coordinate, and this is related with one of the first things I said yesterday morning, that is we want to know what are the important variables in our system. Okay. Yeah. I just keep one of the 20 topics. Okay.
That's good. Really right.
So yeah, this is related with the idea that we need to know what are the slow variables, the important variables describing a simple a process and that there is no trivial way to know it. And this is true at different time scales. So in the longest possible time scale, this is the reaction coordinate. In the longest possible time scale, for instance, if there is one single barrier in the problem, the diffusion over the barrier, we assume is regulated by one or a few global parameters, and this is what will give you the reaction coordinate. So, and this will also give you the intrinsic resolution of the problem at the slowest possible time scale. This is, I don't want to go into the theory of this, but it's possible to define this problem in rigorous mathematical terms. In terms of more chemical terms, for instance, this is for a very simple reaction, the exchange, hydrogen exchange with hydrogen and heavy water. Even for such a simple reaction, it's not as trivial to pick the reaction coordinate. You have many different choices. The distance of the hydrogen from the oxygen, the distance of the deuterium from the oxygen, or the distance of the deuterium from the oxygen from the deuterium. And you get different free energy profiles fitting the different coordinates. In the terms of protein folding, we have many different configurations. We cannot even start to define single atomic coordinate as possible candidates for reaction coordinate. We need something that is more global, something that can identify the progress of the reaction from reactants to product, and identify the most probable route from the reaction to the product. So we have a bag of coordinates, and we like to have a way to, and these coordinates can be given by experiments or by molecular dynamics simulation. Just that this very large set of configurations, we want to find a way to order them so that we know how they are populated along with the reaction. Clearly, this is a diffusion process, it's not a deterministic process, but we want to have a probability flux from one region to the other. So, and this is the figure I stolen from a recent paper in Peter Wallen's group. So we want, if we have this coordinate, we can order all our configurations in protein folding reactions such that if you follow a trajectory, if you have a particularly smooth landscape, you'll have that the system spends a lot of time here, and then at a certain point there is a real event, it goes over the landscape and goes to the other side. But also if there is a more rugged landscape with some more frustration, it can happen that there may be recrossing, but still the probability, the diffusion here is much faster than the diffusion here. So, so that the system spends more time, much more time here than here. Yes?
Because the W is not moving out.
No, this is just the way it works. So the system spends a lot of time in this basin, and then there is a real event, a real fluctuation that can take it on the other side, and so forth. Okay. This if the one single reaction coordinate exists and this can be done, this is what you expect. Okay. You have a question? This is just a noise event.
Just wrong.
Yeah, I know. And if I put it here, then say that I agree. So, there's been proposed that the best way to study this problem is with the so-called P-fold, or commitment probability. Meaning, you can define, in principle, for each structure, a given particular temperature and a given energetic model, the probability starting from that structure, if you start an infinite number of trajectories, the probability of visiting the transition, the folded state before visiting the unfolded state. You can imagine that you have one structure and you start a very large number of trajectories from that structure with random condition at a given temperature, and you count how many times you go to the folded state or how many times you go to the unfolded state. And when you reach the folded and unfolded state, you stop the state. So, and the idea is if you are exactly on the top of the bar, you have 50-50 probability. So this is the perfect reaction coordinate because it identifies the transition state in a rigorous mathematical way. The problem is that doesn't work. First of all, it doesn't work practically because you cannot, you run a simulation and you collect a set of structure, and then you have to run an infinite amount of simulation for each structure, so you never compare. So practically, only people who have folding at home can do it. So you can imagine who invented this reaction coordinate. And also...
The worst part is you have any minor roughness.
Yeah.
No,
wait, I'm going to tell you a story. So, when last year I was with Jose, we sent this paper, and the referee, that was a completely different topic, and the referee asked us that the paper could not be published if we didn't do the default analysis on all our structure. So I spent the full summer before to move to Houston to do default analysis on all the structures for this simulation, and this is the result. So this is one particular trajectory. This is the folding of one circular predicate cube. So this is the reaction coordinate cube. This is a completely unfolded structure, and you observe this transition for the structure, and then unfolding and so on. And this is what I mentioned yesterday when I said that with this simplified model, you observe a lot of folding and unfolding transition because they're very fast. And we pick the top of the barrier and we look at all the configurations that fall in this region that are good candidates, because this is corresponding to the transition state, at the top of the barrier. These are good candidates for the transition state. We divide them in different groups according to the fact if they are in a productive route from the unfolding to the folded, or in an unfolding event from the folded to the unfolding, or they are just recrossing, is it just an attempt to fold and then it goes back, the attempt is and then it goes back one direction or the other. And the thing is that if we look at the probability of forming particular structures in the 4 different groups, it correlates very well. So these are the probability of single contact computed in these 4 different sets, meaning that there is only one transition state in some way. Cumulative transition state in some way. It's not that you visit 4 different. Not that this is a trap. It's the same set of structure as this one. And then we did the full fold analysis, and I hope I'll never have to do it again in my life. And this is the result. So it's a very bimodal. So for this protein, there is a 50-50. For all these structures here, there is a 50-50 probability. Of course, depends on the temperature because it can tilt the landscape one direction or the other. So we did it for 3 different temperatures around the folding temperature, and we find the 50-50 probability for a region, for a Q around what we indicated as top of the barrier. So at least in this, I mean, when we could do the a posteriori test, we cannot use it a priori as a predictive reaction coordinate. As a very unknown a posteriori test, we can do it, and it takes 3 months, but we can do it, and it seems to work. The worst thing is that p-fold, so you can Q this overall global empirical reaction coordinate seems to agree with the p-fold for simple cases, but the problem is that p-fold is not a good reaction coordinate for more complex landscapes. That is, I've taken these from a recent paper from Peter Warren's group. So you can imagine a scenario where, the pictures I showed you, the definition of the transition probability, if you have a single barrier, okay, you have 50-50, you have a transition state. But what if there are 2 barriers? What if there are multiple routes? What is P going to tell, P4 going to tell you? It can happen that the probability, the 50% probability is in a local minimum between 2 barriers. And this is what happens in this case. This is these black points are the default analysis, and the default 50%, if you use Q, you see that you have two barriers and the minimum in between. And if you use two reaction coordinates, you see that this is the minimum that is visited in between the folded and unfolded state, and this is what before we pick as the transition state. And I mean, there are many examples in this paper. So, because default is just one parameter. If the folded landscape is not intrinsically one-dimensional, we don't know what to pick. And there is no way to know just using default a priori if it's intrinsically one-dimensional. But on the other hand, it's not very convenient to start to try many different sets of empirical reaction coordinates. You can get lucky and you can get a very good idea, but you would like to have a more robust way to identify, first of all, the intrinsic dimensionality of how many coordinates you need, and what is the best definition. So this is, again, a work in progress. We have some results that I'm going to show you, but now we have a collaboration with mathematicians in order to make it much more rigorous from the mathematical physics point of view. So the idea is the following. We want, as I said, we want a way, a technique to take a bag of configurations and order them in a way that gives us a probability flux from one very probable point to another very probable point. This problem is very similar to a range of completely different problems in other fields. For instance, image analysis and recognition. If you have one face, or if you have one picture, like this, you have a certain number of pixels. In this case, it's 64 by 64 pixels. If you have two pictures, both of them same number of pixels, how do you compare them? How do you know if it's the picture of the same person? Clearly, this is, in the United States, is very important for national security, so there is a lot of work done on this in recent years. You cannot compare pixel by pixel. If you compare two pictures pixel by pixel, you never recognize the same face, because the color can change, the orientation of the person can change. You want some global parameters to describe as these picture changes that are not too depending on the single pixel. So again, it's the same idea. We want reaction coordinate to compare the essential features. In this case, for this particular, if you could do it for this particular set of pictures, you could order all the set of pictures in this way, the same space, that only changes two parameters, the up-down pose, the left and right pose, and the lightning direction. So only three features to discriminate, to identify, to organize all these pictures that seemingly are 64 by 64 dimensions. We want to do the same thing for our processing system, and so we started to look at the literature in this respect, and we found something very interesting. So mathematically, this problem is the problem of finding the real dimensionality, as I mentioned. What does it mean? Imagine that you have a set of points, and you are given this set of points, a set of coordinates, but if you could plot these points, they live in a submanifold. For instance, in this case, if I give you each x, y, z of all these points here, you think, just looking at x, y, z, you think that these points live in a two-dimensional space. But when you plot them, they are nicely all lying on a torus. So this is a two-dimensional surface. It's just that I'm giving you the wrong parameterization. So what we do when we look for a reaction coordinate, we assume something similar. We assume that there is some global parameters that can parameterize, can span the whole space, that is the whole relevant space that is visited by the protein during the folding transition. So when we use the x, y, z coordinate of each single atom, we're just using your own parameterization. And we know that information is redundant anyway, because we know that there are constraints, there are bonds, and so on. And we know that there is cooperativity. So one way to explore the geometry, so what we want to do, we want to explore the geometry of the space where the points live in. And the way to do that is a tool in differential geometry is, if you can define geodesics on on a manifold, you know everything about the manifold. So the geodesics between 2 points is the shortest path on the manifold, on the surface, on the digital surface, connecting the 2 points. So for instance, if I have 2 points on a sphere, this is the shortest path between these 2 points. If these 2 points are on a torus, this is the shortest path. If you have a way to define geodesics between any pair of points, you know everything about the manifold. You know the complete geometry of the manifold. So you know what is, in implicit way, you know what are your points within and all the geometrical properties. So we took this idea from a computer science paper. We can approximate geodesics, assuming that we have a very good sampling, we can approximate geodesics between 2 points as, first of all, we do a nearest neighbor network, yes?
So you assume there's a general manifold that can
You get a microphone so suddenly you stay.
So you basically assume there's a general manifold that you can reparameterize your reaction space, which gives you better result. But what is the reality behind these coordinate transformations that you can assume? Make the assumption. And also like with different protein, with different molecules.
I'm describing the general principle. If I'm saying if there are the ideal reaction coordinates, this ideal reaction coordinate should span this manifold. I'm not telling you that the manifold is in a particular shape. I'm recasting the problem in mathematical terms.
Would it be like turn out it would be like you reconstruct manifold to fit into your problem?
Yeah, this is what I'm doing. But I have to do it using the proper mathematical and geometrical tools. So what I'm telling you, in order to reconstruct a manifold, in order to know everything about the geometry of the manifold, one way is to investigate the geodesics. Because the geodesics is equivalent to the magnitude of the manifold. So a way I can reconstruct geodesics is that I assume if I have a very dense sampling, I assume that if I have a smooth landscape, and I assume that the landscape is smooth because I know the minimum frustration principle and everything reasonably smooth, everything is linear. So locally I can approximate any manifold in an epsilon neighborhood. I can always approximate a manifold by a Hilbert plane. So that means that locally I can use Euclidean distances.
But you can have infinite number of manifolds that fit your column.
No, that would be defined. Because it means that they don't lie on a submanifold. So, yeah, so in order, using this approximation, locally I can define Euclidean distances and I can hope through a nearest neighbor network to define the manifold, and then I can use this to learn about the property of the space where my point is living. This work was done, a student pioneered us in collaboration with a computer science student, Nan Schematic, who was also the art one of the student I told you before. All the students in my group are somehow related. And this is the application of this idea to the folding of SH3, the landscape I presented yesterday. If you take all the points, and I think overall we took something like 6 million points, 6 million structures sampled through multiple folding and unfolding simulations for these proteins, and we use this method. This is what we obtain. One of the nice properties of this method is that you get an estimate of the error. The residual variance is the error you make if you take only 1 or 2 or 3 or 4 and so on coordinates to span the space of your point. So if the residual variance is 0, it means that you can exactly reproduce all your points using only these parameters. And this is a comparison with a linear dimensionality reduction, popular linear dimensionality reduction tool that is principal component analysis, that for Totem folding fails miserably because folding landscape are not linear. So this is why we need to use the basic idea. And you see that the agreement is pretty good, and what it's saying is that the error goes to 0 after 3 coordinates. So you need only 3 parameters to span the overall landscape of this protein. So it's intrinsically, this means that on the longest time scale is intrinsically a dimensional process. So this is very good news for theorists because it means that we can indeed define these reaction coordinates. And if we look at the first one, that is already pretty good because already the error in using only one coordinate is pretty reduced. It's very similar to Q. There is a very large correlation with Q. The transition state is given by this coordinate is the same as this empirical parameter Q, because this is a well-behaving protein. And so this is what we get. We use now this first coordinate, we call it first embedded dimension, to plot the free energy landscape, and we find a nice single barrier process. Then we take all the points on top of the barrier, and we did the p-fold analysis again, but this time I didn't do it myself, I asked the student to do it. And this is, we get an extremely good correlation, 99%, so indeed we find the transition state. This seems to be a very good coordinate. There is some variation around the correlation because we need to, the error is not 0 using one reaction coordinate. If we use 2 reaction coordinates, we do a much better job. Here we do the default analysis over the
If I ask you a couple of questions here, describe. I know with 2 you're going to do better than 1, but can you show us a case where you really need 2, then 1 will fail pathetically? And second, you have one case where the first coordinate is not related to Q for some reason.
Yes, I don't have data for that one here yet, but yes. For instance, for photoactive yellow protein, the Q1P, I'm analyzing it, but we need, this is not very useful, we need other tools to do that. But I have, for instance, we've done a very small analysis for the iron peptide, but it's short peptide. In that case, you need 2 coordinates because it's a frustrated system. If you have frustration,
frustration you need 2. But also, actually, you may find some system that's not Q, is a sort of weighted Q.
Yeah.
But they're not going to go too far out. There's not too many choice for the first one.
Yeah. But it's nice that for systems where we know that we're behaving, the first coordinate is in agreement with 2. And this is to put the 3 of them together, 3 coordinates, and we get this 3-dimensional landscape. So this is clear. This is what we want to have, this flux from the unfolded to the folded state going through the transition state. And so this direction here is Q, is the first coordinate that gives the folding unfolding direction. Then there is the second direction is the fluctuations in the unfolded state. And the third direction is the fluctuation in the folded state. And we can group all the structures. So each point here is an ensemble, and there is and we can look at the structures and the evolution of the structure as they move in this. So we can represent the landscape, the folding landscape, in a low-dimensional way. Now, this, as I said, this is just the beginning of the story because it turns out I didn't mention energy in any of this. We need to weigh all of this with an energy factor, and we are collaborating with a mathematician, and we managed to write these in terms of diagonalizing the Langevin operator. So this is becoming a very rigorous tool. So just to conclude, I want to thank all the people who are working with me, or have been working with me. This is my current group. 3 of them are here, and these are past group members, and all the people are giving me money, and my collaborators, first and foremost, Giovanni Costati, my husband, and also my computer administration guy, and all the people at RISE are collaborating with me. Thank you very much.
We have some time for questions.
This might be obvious and I've just missed it. For when you do the geodesic analysis for 1, 2, 3 important parameters, does that tell you something about the roughness in each coordinate?
No. Well, yes. At this level, no. But the next level that was okay, this is just the beginning. The next level, we have a neighborhood that can expand and contract, and this is related with the roughness. So we have a parameter that tells us how good is the linear approximation locally. If the locally you are restricted in a very small neighborhood for the linear approximation, means that you have a rough component.
So what she's asking is, I agree with your linear approximation, but how small has to be the region? Basically, if you increase the roughness, that region becomes so smaller, that can make your life. But how do you determine a priori how small that region has to be? And I think that's what he was trying to address before.
So it's not, I mean, I don't have a very direct answer. We are using tools from harmonic analysis and the eigenvalue, the composition of the Laplacian operator. In this, we can in kernel basically, you look at diffusion locally and you study the properties of the diffusion locally, and you can look at how good is the linear approximation.
So you have to have a iterative method that you go.
Yes. So yeah, we know that we can resample in different regions.
Here's our guy. He likes multiple. He's going to tell you there's not a single solution.
I just have a very simple question. So when you map your points into the manifold, you first write down the metric, right? So what number tell you whether this is a good metric for the conformal map? You embed.
So yeah, the number is this receiver variance. This receiver variance is the error you make when you try, and it's normalized between 0 and 1. It's the error you make when you try to reproduce all the points in your manifold using only 1, 2, or 3. It's a function of the number of coordinates you use. So if you use only one coordinate, you already do a very good job because you make an error that is more than 1% in reproducing the points in your manifold on average. If you use 2, you go below 0.5%. 3 is below 0.2%. And then, okay. And this is the similar error we make if we have to use principal component analysis. Principal component analysis, the first dimension doesn't tell you anything. Because if you have a nonlinear manifold, if you have, for instance, a torus that is very nonlinear, if you project this on a plane, this is what principal component analysis does. You distort everything. You mix together things that are not together.
Okay, do we have any more questions? No, that's thank you to see you. A few things more to tell people. As you go out, there is an evaluation form that you're actually also getting by email. But based on my experience, I would appreciate if people fill this up and turn it before you leave today. The chances, if you say, oh, I do that later, not to do it, increase exponentially with time. So also your brain is fresh. A large amount of this program, both in the US by NSF and here in Rio, where FAPERJ is supporting, we are stronger believers that based international cooperation, that bringing people from different countries together is a great thing. But we need support for the people, their students, that actually tell that they like it and what the situation is. So when you get this paper, there are several multiple choice questions, but there's something here where say any additional comments you have. This section is particularly important. That sort of gives some meat when people prepare renewal of these proposals and stuff like that. Also feel free to put some suggestions of what could be better, what could be worse, what could be done. So feel free to do these things. So the forms will be given to you outside right now. So I really ask that you do your best job on that. One more thing, as you notice, today is Holy Friday, so let's pray. No, I'm joking. So everything's closed, as you can see. But I think there's, in the shopping mall, there's some restaurants open, right? Or at least some fast food place. That's what Lisa told me. I don't know, I don't know.
Yeah, no, I think it's open.
Yes. So real soon there, you must have at least some things like McDonald's will be open. But anyway. No. Aren't you Italian? Don't you know today is Holy Friday? You're supposed to fast. Don't you know that? Okay. Do I have an Italian? What's going on here? Okay. So Alex is giving our final 2 lectures, so you're going to finish with a golden finish. So everybody here at 1:30, okay? Bye-bye. Pick up the graduation on site.
Make a continuation, leave the break to the end. But I think it is too much for me. So we are going to keep the same exchange.
Okay, I can't express my gratitude enough that you actually found the strength to come at this last afternoon, especially because the sun at Praia Vermelho is so nice, it's nearby. So I'll try to make it as entertaining as possible. But some of the things I'm going to discuss are pretty hard, so we'll see how it goes. So I wanted to start today with telling you about the second problem associated with mitotic spindle. And this problem is mitotic checkpoint mechanism. So this is one of the, probably the most important thing in whole mitosis. The most important point at which mistakes are most lethal for the cell. And here's what I mean. So when the cell is at this stage, at the end of the prometaphase, beginning of the metaphase, when most of the sister chromatids, most of the chromosomes are captured from opposite poles, and hopefully all the attachments are correct. We discussed yesterday this incorrect neurophilic attachments. But here's the question. So some of the chromosomes will be captured early, some will be captured later. So the cell has to wait until the last of the kinetochores, this tiny organelles, tiny droplet of glue, until the last one is captured from the proper pole. It has to wait for this before it starts dissolving the glue between sister chromatids, and before it allows in anaphase, it allows molecular motors to pull the sister chromatids to opposite poles. So it shouldn't start pulling and dissolving the glue between the sister chromatids too early. If the mistake made of this sort, this is lethal, this is cancer, death for sure. So how does the cell do it? There's a very interesting mechanism which is one of the examples of this mechanochemical mechanism. So I'll first tell you sort of in broad outline what that is, and then in the end we'll discuss what the molecular structure is. So here's the idea. Each of the kinetochores, each of the unattached kinetochores, trigger the sequence of chemical reactions. You'll see in the next few slides the whole set of reactions. But for now, sort of the primitive view is this. On the kinetochore, molecular complexes get activated. And here, just 4 of the main player proteins are shown. In fact, there are more. It's close to 20 known proteins participating in this set of reactions. So this complex gets activated. Okay, I'm not going into molecular details of what does it mean to get activated. It means phosphorylating some of these proteins and dephosphorylating some of them, or some others. So then this complex binds to enormous protein complex called APC, standing for anaphase promoting complex. So APC is this huge machine, kind of, that can be compared to ribosome in complexity. So when it's inactivated, it doesn't do anything, so the cell just stays in this state. However, when the last kinetochore has microtubules attached to it, right, so the spindle is completely assembled and ready to segregate chromosomes, then there's no more unattached kinetochores. So this complex is not getting activated, and inhibition on APC is removed. And then what anaphase promoting complex does, it takes the famous protein called ubiquitin and passes it on these droplets of glue between the sister chromatids. So let me actually show it to you on the next slide. So here's sort of what happens. So here only one out of this huge complex of proteins is shown, called MAD2. So it stands for mitosis-associated disaster or something. It's not because it's a crazy protein. So when kinetochore is not associated, not yet attached, this MAD2 is going to get activated on the kinetochore, right? And then it inhibits anaphase promoting complex together with another important protein called CDC20. When microtubule attaches to this kinetochore, so MAD2 cannot physically get access to the kinetochore anymore. So MAD2 is not getting activated, and it stops inhibiting this complex. So then APC, in complex with podosome protein, takes this ubiquitin chain and passes it on many different things, among them onto this glue between sister chromatids. And you might maybe learned before, if you haven't heard before, when ubiquitin is attaching to any protein, this is kind of mark of death. This protein with ubiquitin on it is ready to get proteolized, to get destroyed. So here are a couple of very important experimental facts. So in the middle of the 90s, a very interesting experiment was done when two cells were fused. So two cells now are sharing two spindles. Okay? And interestingly enough, in one of them, right, there was unattached chromosomes, and so there was wait, don't do anaphase signal in this spindle. But the other spindle was going into anaphase fine. Okay. So it means that so the signal which I'm describing, this one, has to be diffusible, right? Because if it wasn't diffusible, so this kinetochore should send a signal to every other chromosome. So the signal should somehow get around in the whole nucleus. But at the same time, what this experiment is saying is that the signal shouldn't diffuse too far. It should die out relatively soon in the cytoplasm. And there's another hint that this actually happens because with one of these FRAP techniques, the half-life of activated MAD protein was measured, and it stays in activated state only for about 20-25 seconds, which is very short for some characteristic protein in the cytoplasm. So the idea now is that probably what happens, MAD is activated and then diffuses, floats in the cytoplasm, but then gets deactivated really soon. And remember, we discussed last time this RAN protein making smelly cloud around the chromosome. So that's probably something similar going on here. So MAD2 star creates kind of a finite size cloud which includes all other chromosomes, but don't go past immediate spindle. Okay. So there's something very elegant and controlled going on here. So here are three interesting questions. What is actually the composition of all the reactions? So I described sort of crudely what's going on, but what are all the reactions? How do they go? Second, there are actually, you can imagine, there are two requirements for this mitotic checkpoint. One requirement is that just one unattached kinetochore should maintain tight control over all other kinetochores. So it shouldn't be loose, so it shouldn't make mistakes allowing other sister chromatids to start separating. At the same time, when the last kinetochore is getting attached, this inhibition should be removed from the whole spindle, and it should be done fast, because the cell already wasted too much time on spindle assembly, on all other processes. It can't wait, it has to go into anaphase. So you'll see that those two requirements, 2 and 3, are contradictory. You can't at the same time maintain too tight control over the whole spindle and remove this inhibition fast. Okay, so again, it's the same theme which permeates the whole cell biology. The cell has to do mutually competitive, mutually exclusive topics.
So I'm just going to ask, what are the timescales for what's the time difference between the completion of the last chromosome and the pairing of the microtubule of the last kinetic core and the onset of mitosis? What is that timescale?
So it's a few tens of seconds, depending on the cell. For yeast cell, which I take the data from, it's about 20-25 seconds, which is very fast. So first, let me show you a little bit about answer to the first question. So the next few slides will look very scary, but don't worry, because you don't have to memorize all those details. So I really urge you to, people who are interested in sort of biophysics of on the cell level, to read this paper. I'll discuss it in this and next few slides. And I think it's a bad paper, but other people are of different opinion. In any case, it's a very educational paper. It's worth reading, and I'll tell you why. So so this is a completely theoretical paper, and it basically mathematically simulates the bunch of chemical reactions. So first of all, what's very educational in it? Work like that is very popular these days in quantitative biology, and technically, nothing is easier. So what is shown here is fashionably called in system biology these days is interactome. Okay, so what does it mean? Well, all these different species are molecules participating in mitotic checkpoint. And decades and hundreds and maybe thousands of papers from experimental biology and biochemistry went into identifying all these players first, and then figuring out which who is involved with who in chemical reactions. So all these lines with arrows means that certain reactions takes place. For example, what's the simpler one here? For example, here this huge complex with attached CDC. What can happen? CDC can be removed, this reaction, then this complex can associate with another complex, MCC, get dissociated, and so on. And don't worry about all the different colors, I wouldn't go into these details. So, interactomes like that are actually nowadays known for pretty much any biological process. And this is one of the simplest ones, in fact. Normally, interactome of characteristic, so another name for it is signal transduction pathway. It's sort of set of chemical reactions which have some input, in this case from unattached kinetic core, and output, it is telling the spindle not to do something. So this is one of the simplest. Normally, there are like tens, hundreds of reactions. Here's what is known from experimental biological literature. Now, let me just tell you a little bit about what this is. So this is protein complex consisting of two mad proteins attaching to the kinetic core, and they are not activated yet. Then another mad protein comes, associates with this complex, makes this bigger complex. Then this bigger complex takes CDC, one of the important proteins, gets CDC on board and loses a big chunk of initial complex and get this complex. So this is activated MAD. Okay. Then this important complex get associated with another complex made of so-called BAB proteins. It's also the name BAB also comes from some abbreviation, but I forgot what it is. So together they make this complex that is called MCC. So only this complex can now inhibit APC, which is really huge, and which is ready to give a signal to go into anaphase, but doesn't, because MCC associated with APC inhibits APC. So when last kinetic core attaches, U means that when kinetic core attaches, those reactions get cancelled. So where you have U, those reactions reactions are cancelled, and basically the result is that now almost no such complexes exist, and APC freely associates with CDC, and this one is active. It makes the signal to go into another phase. Okay, so qualitatively, it's actually very, very simple. There's no big mystery here. So here's the interesting question that I want to pose at this point. As I said, it's not too complicated, but still, you know, it uses more than 10 proteins and more than 10 reactions. Why do all this stuff? It shouldn't be just a couple of reactions. You can easily achieve the same goal that I described with just 2 reactions. Okay? So very often in cell biology, you will hear this argument that all these other reactions are so-called garbage of evolution. You know, so the cell, of course, wasn't designed by a divine being, though there are arguments to the contrary, but developed this whole stuff in the process of evolution. So many errors were made in evolution, right? So the idea that some of those errors didn't die, they just exist as useless reactions in the cell. But many people, myself including, are of different opinion that all garbage reactions, well, I am not an expert, for example, in genetic regulation networks, but I know a lot about signal transduction on biochemical regulation networks. I don't think there are any wasteful reactions there. So all these reactions are needed for something. And by the end of the lecture, I want to give you a hint what for do we have so many reactions. Okay? There's no answer yet, but there are some glimpses of what the answer is. So now, what do you do when you have this interactome? Then, of course, you need to know all the concentrations and all these numbers, all the coefficients. So what the authors did, so first you just write schematic reaction. So let's look at the simplest reaction, which is one of the last. You have APC by itself associating with CDC, right? So corresponding rate of reaction is K8. So the association and dissociation with rate K minus 8. It's really nothing. Any undergraduate student of the first year can do it. How do you write corresponding differential equation for this reaction? Well, just like that. Derivative of the concentration of this complex, right, is minus K minus 8 times this concentration plus K8 times product of these two concentrations. As easy as that. Of course, behind it is a fundamental assumption, which is called mass action law. No doubt all of you have heard about it, which tells you that if two molecules are associating, then the rate of reaction should be proportional to the product of these two concentrations, which is far from obvious. Many reactions are not like that. So this is a huge assumption this paper makes, that all these reactions obey mass action law. It's not clear, but they make this assumption. And second, all these coefficients are gathered from huge biological literature. So this is my first criticism of this paper. Normally, these days, people are very impressed if there's this table in the paper. So all of these coefficients are known. But if you read carefully, all of these concentrations were gathered, of course, from different experiments, most importantly from different organisms, different conditions. Okay. So there's no guarantee that it all will work like that in a single cell. So again, it's good. It's not a bad paper, but you always need disclosure. You always need to say that this is the caveat, which these authors do not say. And let me comment also when I told that this is very easy work technically, why I said that. So if you, of course, start writing by hand all these equations and start writing MATLAB code, let's say, by yourself, it's easy, but it's pain in the neck. It takes time. It's very easy to make a mistake, then debugging the code. So why it is easy these days? That there's lots of excellent software around. All you have to do there is to sort of draw boxes, each box for each molecule and molecular complex. Make lines between these boxes and assign numbers to the lines. Okay, there's and then computer will write equations for you and solve them for you, and choose the ODE solver for you. So what I want to emphasize, if you think of seriously working in theoretical physics, you have to start paying attention to all this software, because in the past, people, including myself, were very contemptuous of this friendly software, because like serious physicists should write his or her own code. Okay, so the situation is not that simple anymore, because again, you waste too much time writing it yourself, and also if you are concerned only with time dynamics, there's just no reason to do it by yourself. You have to use one of those codes. And there's also a great advantage of doing it, because, for example, what is it, CDML or something, some of those codes are now designed so that they can talk to any other software. They can talk to MATLAB codes, to Mathematica codes, to Pascal, whatever. Okay? And you can immediately share your model with everybody. You can take anybody's model. So it's a must, and also it's fun. It's good stuff. Okay, so after, what does people do? After they encode all this stuff with equations like that, they gather all the parameters, then they solve it, and solving it is just punching a button. You don't worry about how to do that. And here's the example of the solution. So up to 2000 seconds, they simulate, so u is equal to 1, meaning that there's weight anaphase signal. And they solve system of equation with arbitrary initial condition, and they got steady state. So let me just concentrate on MCC APC. This is inhibited APC. So it's high, right? So the cell wouldn't go into anaphase. And then at this point, last kinetochore gets attached. And then they make u equal to 0. Some reactions are getting cancelled in this interactive electrode. And you see that inhibited APC drops. That's it. Okay, and there's new steady state for everything. So here's my criticism of this result. It's very nice, but in fact, you could explain all of that without solving equations qualitatively. So that's what biologists sometimes call slapping differential equations onto something which is qualitatively already clear. Okay, so it's a good thing to do, but it's not very revelatory. Because all this transitory time dynamics is not a big deal. Well, they show that everything happens within 500 seconds, actually smaller. This is not important, something like a few tenths of seconds. But it's already clear from when you measure the rates of reactions. There's one really good stuff in this paper, actually. They simulate what happens in about 10 known mutants of yeast cell and biochemical inhibitions. And for example, in one of the mutants, when there are no MAT2, well, no surprise, the anaphase never will happen. So there always will be a weight signal. So the concentration of inhibited APC is always high. In another experiment, in overexpression of MAT2, there are some different steady states. And they kind of qualitatively compare it to the known measurement and see good qualitative correspondence. So again, I don't want to criticize it too much. All I'm saying is that part of this model is kind of, it's not a fantasy, but it's, they know what they want, and they kind of pull coefficients that they want. They sort of, I think, such models are a little bit, there's a colloquial expression, they're rigged. So they are designed to do certain things. But it's still a good first step. Now, let me also emphasize one interesting thing. I will tell you later about our own work, and we use a similar trick. So, not all of the parameters in this model are known. 4 parameters were never measured. So what they do, so the problem with the modeling when you don't have parameters, right? If you have even 2 or 3 unknown parameters, with enough equation, you can predict anything. Right? So there was this famous Eugen Wigner expression. For those of you who don't know, Eugen Wigner is one of the founding fathers of quantum mechanics, Nobel laureate, real heavyweight. So he was saying, with 4 parameters I can fit an elephant, and with 5 I can make it wiggle its trunk. He was referring to this method of approximating shapes with splines. But that's true with modeling in cell biology. So what these people do is very smart. They actually had some experimental data, quantitative data, and they use this data to calibrate these parameters, to find these 4 parameters. Okay, so now on this work, I wanted to tell you about 3 other papers, which I think are way better. Especially, I totally admire this paper. I just love this. I really recommend, for those of you who are interested in it, to read this. So what they do is, they do exactly what physics is good for. When you don't know all the details about the reactions and what's going on, you still can say something very useful if you know what question to ask. So this is a group of Nama Barkai in, I think, in Weizmann Institute, and she was not founding father, she was one of the founding mothers of modern quantitative system biology. So here's what they ask. They're not trying to model this big system of reactions, which anyway is not completely clear that this system of reactions is true, actually. They're asking interesting question. How does the cell do 2 things? So let's look at this. This is sort of caricature of this cell, and one unattached kinetochore is at the center. So there's this unattached kinetochore. You can imagine it's kind of in the center of the cell. So it is sending a signal which has to be felt in the whole nucleus that there shouldn't be anaphase. So think about what's happening. You are taking one molecule which is floating in the cytoplasm. You're activating it on this kinetochore, and then this molecule floats away, but it gets deactivated anywhere. So far enough from this kinetochore, the molecule is deactivated. So far enough from this kinetochore, you cannot maintain this signal not to go into anaphase. And experiments show that too far from it, the signal doesn't work. But so there should be strong enough signal in space, right? So percentage of activity should be low enough, far away from this kinetochore. At the same time, when this last kinetochore gets attached, you have to remove